Whenever we look at these little components called organelles, we usually end up looking at the nucleus first. The nucleus houses all of the DNA of the cell. And DNA can come in many forms. And we know that one of the forms that it comes in is this double helix type structure that I'm poorly drawing right there. But the DNA also comes in other forms like chromatin, chromosomes, and other smaller divisions. But no matter how you refer to it, it's always found in the nucleus. If your DNA is not in your nuclei, you should see a doctor. The nucleus, it is a double wrapped membrane, and it has what we call the nuclear envelope. Is this a cell membrane? No, doesn't have cholesterol and some of those other components. It's just like a plasma membrane with only these little openings right here that are called pores. None of the other stuff, like no proteins, no cholesterol. The only things that can come into and out of the nucleus, and the only things that have the right shape to pass through these pores, are nucleotides of DNA, nucleotides of RNA, mRNA, and tRNA, all of which will be explained in the future. Red blood cells, they have no nucleus. They threw it up or exocytosed it long ago to make more room for oxygen. On the other hand, white blood cells, they have a huge nucleus that grows during your lifetime. The nucleus is making memories, much like your brain makes memories. Your brain rearranges your neurons to convert short-term to long-term memory, which, by the way, takes sleep. White blood cells, what they do is they make DNA to remember the invader or the pathogen and remember how to fight it. So after you got vaccines for things like measles, mumps, rubella, chickenpox, COVID, some of your white blood cells recorded how to fight those invaders. What is an endoplasmic reticulum other than something that looks like a maze of tubules? So let's just do some root word exercises here before we get into the organelle and what it does. Endo, you know what that means. It means inside. Plasmic, well, we must be referring to something that's inside the plasma membrane. And a reticulum is a network. The word means webbing, netting, or network. And that's what an endoplasmic reticulum looks like. We're going to consider the rough and the smooth endoplasmic reticulum, and we're gonna abbreviate both to be ER. But the SER and the RER, they are very separate organelles, even though they look alike and even though they're attached to each other. The smooth ER is a lipid-making organelle. Its main role is detoxification. It makes lipids that basically surround toxins, allowing their removal. So your liver cells, called hepatocytes, they filter toxins from your blood, and they tend to have lots of smooth ER. Alcoholics with a tolerance to alcohol have tons of smooth ER in their hepatocytes as they're trying to eliminate all of the excess alcohol. Cirrhosis of the liver, which just means inflammation of the liver, it can result from the overabundance of smooth ER. The rough ER has many jobs, but most of them have to do with building a protein, like folding it, packaging, well, folding it. The rough ER plays a role in folding the polypeptide chain, and it can also make a little bubble of phospholipids that's called a vesicle, or a transport vesicle. And that bubble or vesicle, it contains a protein, so it can be transported to the next organelle in the process of making a protein. But the main difference between these organelles is that the smooth ER gets a lot of dates. Sorry, because it's smooth, right? It's really smooth. It's like, hey, what's your sign? Does anybody use that anymore to pick people up? All right, moving on. The Golgi apparatus, it's been called many different names. I think right now I might teach from 3 books that call it 3 things. So I'm just going to call it the Golgi, but you may see it referred to as the Golgi body, Golgi complex, the old Golgi apparatus, but it's all the same thing. I like this organelle because it actually does, like not just in diagrams, but in microscopic pictures, it looks like you stacked up a bunch of pita bread and then cut it and looked at it from the side. So think of how this organelle, it can be a conveyor belt, a protein that's contained in a vesicle from the rough ER. That protein and its vesicle, it enters on this like receiving side of the Golgi body, and that's the side that's facing the endoplasmic reticulum. And then that vesicle, the protein, it kind of gets labeled for its final destination, right? So the Golgi body packages it up, puts a shipping label on it, and says, this is where you're supposed to go. And what the Golgi body does is it pops that protein out in a vesicle that then will fuse with the plasma membrane and exocytose all of its contents out into the interstitial fluids. That's an arrow I'm trying to draw. My pen still doesn't work. But the Golgi body, it makes vesicles for other reasons than just transporting proteins. Some vesicles not containing proteins will leave the Golgi and just get inserted into the cell membrane, kind of like replacing it, like piecemeal, like replacing posts on a fence. Some vesicles bubble off from the Golgi body, and then they turn into lysosomes, which I address in another mini lecture. While the recipes or the instructions for proteins are locked up in the nucleus, the machinery for making proteins is far out in the cytoplasm, or the gooey substance of the cell that's inside the cell membrane. You have to think of the nucleus like the reserve materials in a library. And now that I say this, I'm not really sure that many of you have been to the physical library, which is fine. I mean, everything's online now. But reserve materials, they were things that could only be used in the library. They could not be removed from the library. So things like encyclopedias, dictionaries, you can copy the information in the reserve materials and thus get the information out of the library in your copy, but you can't remove the books themselves. The copy is what is called mRNA. And you can see mRNA down here, even though it's not labeled, which I should have. This dark blue strip here is mRNA. It's being read by the ribosome, kind of like reading a book. And the ribosome, what it's going to do is it's going to make a polypeptide chain and protein according to the instructions on the mRNA copy. We go into this process in much more detail in later modules. Ribosomes, though, they can be free floating inside the cells, so there can be ribosomes out here, and these are not drawn to size. Ribosomes are really, really tiny. Or ribosomes can be stuck on this structure here called the endoplasmic reticulum. Free ribosomes are usually making proteins that will be inserted into the cell. But ribosomes that are bound to that structure there, they're usually making proteins that are going to be shipped outside of the cell. Cells with lots and lots of ribosomes are usually cells that make lots of proteins. Melanocytes in your skin, they make the protein or pigment melanin. Pancreatic cells, which are in your pancreas, they make digestive enzymes, and those cells also have a lot of ribosomes.